Ladies and gentlemen, please be seated because in a moment we will move our second agenda. It is international speakers. So please, for all the participants who are outside, please come join us in the PNC ballroom and please have your seat. Thank you. Ladies and gentlemen, we will now in our second agenda, presentation delivery international speakers. And now, here we are connected with Professor Chun Woo, PhD from Rowan University, United States of America. All right. Hello, Professor.
Hello, Professor.
Can you hear us?
Hello.
Hello. All right. Hello, Professor. How are you, sir?
Good. Greeting from the New Jersey.
And greeting from Indonesia, Southeast Sulawesi, Professor. We also deeply thankful for joining us for HICPAS 2023. Today, Professor, you will be our first speakers for international presentation. So how are you feeling, sir, about that? Are you excited or
it's great
maybe
honor.
You can share with us with your feeling?
It's great honor.
I'm sorry, sir?
It's great honor.
Okay, it is also a great honor for us. Maybe we will hold back, sir, and we will come back to you in a few moments. Because before that, I would like to inform for ladies and gentlemen, in this session, we will talk about debunking old evolution theories, establishing new theory using SARS-CoV-2 genomic data, who will be presented virtually by Professor Chun Woo, PhD from Rowan University. In this session, we will be guided by our moderator. Please, invited to the stage, Professor Dr. Muhammad Arba. I would like to introduce the moderator for these sessions. He is a lecturer at Faculty of Pharmacy at Universitas Halu Oleo. His research focus is in the fields of pharmaceutical analysis and medicinal chemistry. His educational history includes a bachelor degree at Universitas Halu Oleo. He completed his master's and doctoral degree at Bandung Institute of Technology. What is interesting about this person has received his professor degree at earliest 40 years old. He has superior research from the Ministry of Research, Technology and Higher Education, and has written many articles published in reputable journal. His complete profile, you can read it on the big screen. Professor Dr. Muhammad Abra, this session is yours.
Okay, thank you, MC. Very nice to be here. Assalamualaikum warahmatullahi wabarakatuh. The Honorable, the head of Education, Development, and Quality Assurance, Universitas Halu Oleo, Pak Dr. Laude Santiaji Bande, the Honorable Dean of Faculty of Pharmacy, Universitas Halu Oleo, Professor Dr. Ruslin, the Honorable Dean of Faculty of Pharmacy UGM, Professor Dr. Satibi, Professor Taiful Mahmud from Oregon State University, and all presenter, oral and poster presenter, all participants, both onliners and offliners. I am Muhammad Arba, will be your moderator for this session. I am very pleased to welcome you in this prestigious event, the first Halu Oleo University International Conference on Pharmacy, Health and Applied Science 2023, held by Faculty of Pharmacy, Universitas Halu Oleo. Ladies and gentlemen, our first invited speaker today is Professor Chun Woo from Rowan University. Professor Woo, can you hear my voice?
Mute. Yes, I can hear.
Okay. Okay, let me read his curriculum vitae. So Professor Woo obtained his bachelor in chemistry in Xiamen University, China, and then he completed his master degree and doctoral degree in University of Delaware, USA. He then pursued his postdoctoral study at the University of California, Santa Barbara in 2025, and then since 2009, become assistant professor at University of California, Santa Barbara. Since 2018, he was appointed as associate professor at Rowan University, Department of Chemistry and Biochemistry, and Molecular and Molecular Bioscience. He is also a member of Beijing Computational Science Research Center in Beijing. His research interest is computer-aided drug design, molecular modeling, and molecular dynamic simulation of biomolecular systems. He has published more than 90 papers and has almost 10,000 citations. So no doubt he is very expert in his field. Today, Professor Wu will deliver his talk about how to develop vaccine and antiviral drugs using the new evolution theory. To be more specific, the title of his talk is Near-Neutral Balanced Selectionist Theory, a quantitative evolution theory to explain genome evolution of SARS-CoV-2 and other deadly viruses and its application to vaccine and drug development. Okay, Professor Wu, you have about 30 minutes, including discussion, and now stage is yours.
Okay, great. Thank you so much. Let me share screen. Hello, everyone. And so this is my office. You kind of can see I'm the computer guy. I really addicted to the computer. And so here's New Jersey in the Atlantic, close to the Atlantic Ocean. And so here's our university, Rowan University, and we just turned to the 100 years old. I really like to thank the Dr. Rushing and also Professor Alba, because we really had a wonderful collaboration during the pandemic. And you see that we had a workshop. And so it's a wonderful experience. I'd like to thank my students. Without their work and also the funding agency, I would not able to present the story to you. Okay, so here's my talk. So let me first start outline of my talk. And so I'll first introduce you mutation selection model for evolution. And then I will discuss two key features of the SARS-CoV-2 genomic evolution. Very interestingly, these two features cannot be explained by existing evolution theory. Therefore, we propose a new evolution theory, and we validated this new theory on a set of deadly viruses, including the SARS-CoV-2. Then we were excited to show the potential application of our new theory in the vaccine antiviral drug development. So let's roll back a little bit about the biology. The evolution, the foundation of evolution is a mutation. Okay, so here I show you the polymerase, which is responsible for making the copy of the genetic information carrier, which are nucleic acid. Okay, so you have this template, and based on the base pairing, this machine will duplicate genetic information. However, it's not perfect, so especially makes mistake. So here are two assumptions about this machinery. It cannot sense position, so you will expect the same error rate at each location. Second, this polymerase is very stable, so you expect its behavior are stable over the time. That basically means the mutation rate does not change over time. And because constant rate, so you expect it. Okay, so we will say the random mutation at the genome and any position, and they all had a similar chance. Okay, and then if we look over the time, because the constant rate, you expect it the linear increase number mutation over the time. So this is based on those two assumptions, what we expected about the mutation. So now let's put this mutation into under the selection pressure. Okay, so here I show you the 3 value, and then they make the replication. So we got 12 copy, and then it make mistake at the beginning, at the middle, and at the terminal, because the no sense position. So beginning, middle, end, they have similar chance. In this case, 3 out of 12 makes a mistake. Okay, we know resources not allow all the variables survive. So only subset variable will be survive. That's basically selection. And then which is selection? The easy selection is no selection, no preference, which are the neutral selection, which basically means each of those variables has an equal chance to be survived here. So here then you will expect it, the population of that mutation will be conserved in here. Okay, so here 3 out of 12, and you conserve here 1 out of 4. So the population, relative population does not change because they all had a chance. So this is called a neutral selection. Okay, with this reference state, now we can interpret what means the positive selection, advantage mutation, and this Darwin calls adaptive mutation. Okay, so look here. Instead of 1, you got 3 that mutant, red mutant exists. And this definitely means it has some advantage to spreading this mutation and fix more in the population. So quantifiably, we can calculate out the ratio. Okay, so the mu is here, and C is here. So in this case, 3 over the 1, so greater than 1. So we had a quantitative measure to show this is under positive selection. And then opposite, here you see that the mutant not able to survive, which basically means it caused damage to the species, and the species not able to survive. What is that type of mutation? Is the disadvantage, purifying mutation. Okay, so how to quantify this one? Okay, 0, you divided by the 1, then this is the smaller than 1. So this is now we got very quantitative measure about the type of selection, type of mutation, used by simply counting the mutation by compare with the random mutation of the replication. Okay, so for neutral mutation, neutral selection, you will expect it because it's a C equal to mu, and mu we know is a time independent. So you will expect a similar behavior you observed before. Linear increase number of mutation, and then you look at the substitution at each position, you should have equal chance to have that substitution. But this is observed in the population. Okay, so with this theory, now let's look at the SARS-CoV-2 we all experienced. And so really what you found, regardless the environment factor change, the genomic mutation rate for the SARS-CoV-2 actually stay constant. That's very surprise to Darwin. Okay, it's not under, appear to not under nature selection. Let's look at the factor change. So this is the 6th wave of the infection we all experienced, right? So you can see the viral population will dramatically change over the time. And this is the vaccination. Roughly 3 years ago, we start to have the first vaccine roll out, and then this line is the person has the first vaccine, this line is the person has the second vaccine, and this line is the person has a booster dose. Okay, so regardless those change, and this is the number of the mutation observed in the genome of the SARS-CoV-2. When we're collecting the sample from the patient, you remember you do the swab, and you see it's a linear increase. So that indicated its substitution rate is constant. And it just looks like the near neutral selection. And then the substitution rate is equal to the random mutation rate, which is 1. And we call this molecular clock feature. Okay, does that mean it's really under the neutral selection? No, it may not necessarily mean it's under neutral selection, because the balanced selection between the positive selection and negative selection also can lead to a near neutral selection. So we have to regularly to check at each position of the genome, are they really have a similar number of the mutation? So this is would be look like here. Okay, and then if we calculate this C over U and to do the population, then it should be expected you have the population which center in the 1. If we saw this, then it's truly under the neutral selection, follow the neutral theory. Okay, so now let's look at what's happening. So this is the genome of SARS-CoV-2. It's coding some the structure gene and some non-structure gene. Okay, so here's what we calculate the relative substitution rate. And remember the 1, and the 1 is a neutral selection. So greater than 1 is positive selection. Smaller than 1 is negative selection. So it's not follow the neutral selection. And then if you calculate the population of the C over U, which is relative substitution, we got this so-called L distribution. And remember, for this L distribution, 1 is boundary. If close to 1 is under the neutral selection. Smaller than 1 is under negative selection. Greater than 1 is under positive selection. So you see, most mutation indeed do damage to SARS-CoV-2, so they're leading to the purifying negative selection. Okay, and this distribution will be not able to explain by the Darwin selection theory. Because Darwin selection theory was suggesting you have the negative selection and then you have the positive selection. Okay, and so we don't have that many positive selection, but we do have some near neutral selection, which original selection theory does not able to explain. And it's not able to explain by the Kimura neutral selection theory. If the neutral, then expect the population around 1, which we don't see that. And it's also not explained by the Alta near neutral theory, because it's expected the near neutral theory will have a slightly weak positive. So this is a new theory we propose. We call it near neutral balanced selectionist theory. So look here, it's the hybrid of the two things, combine the near neutral and also the nature selection theory from Darwin. So that's the new theory we propose. And now we have to explain molecular clock, because it used to be this considered as the feature of the neutral selection. How to explain that? Well, it's a balanced selection. So here you look at, for the black line, is the number mutation in the genome. It's look like neutral. However, if you look into individual gene, and you will see some of the gene under the positive selection, and some gene under negative selection. And when you average them, look like neutral. Okay, so from this diagram, you can see more clear. So this is C over U. Remember, C over U equal to 1 neutral. Greater than 1 positive selection, smaller than 1 negative selection. So you see those genes under positive selection, they basically gain advantage and make this virus more infectious and spreading wider. And those genes are negative selection. Those basically mutation will make it lose function, so it's do bad to the SARS-CoV-2. So based on this, we naming our theory near neutral balanced selectionist theory. Okay, so here's a summary of our theory. So this theory will be hallmark of the L distribution of the relative substitution rate, C over U. And because it's balanced, so if you calculate the genome, we look like the neutral, C over U approximate to 1. But we know it's balanced. You have some gene under positive selection, you have gene under negative selection. Because it is a linear increase, so it can summarize in a set of the simple equation. Then with this one, how this theory differ with the existing theory? Okay, so all the evolution theories say most mutation is bad, so they are under negative selection. Okay, so all the theory differ about what kind of mutation eventually will survive in the population. In the Darwin selection theory, especially say it's adaptive advantage mutation will be survive in population. Okay, yellow one. And in the cumulative neutral theory, say most of the survived mutation are neutral. They'll be there due to good luck. Okay, and alter near neutral say besides the neutral theory, you have some near neutral theory. And our theory say no, there are not much neutral mutation. You only have the near neutral mutation survive in population. And also you have some small percentage of the mutation under strain positive selection. So our theory differ from all existing theory. And our theory now has a quantitative equation describe it. And this work published in January of this year. Okay, so then how to apply this theory? We can identify troublesome gene, and we can determine which gene under positive selection, which gene under negative selection. And then if we already apply the vaccine, we know the gene under positive selection basically would be like the gene has a vaccine escape mutation. Okay, so then we can find out that. And if we already apply the antiviral drug, the gene under positive selection basically would the gene make the mutation, generate drug resistance, so make the virus more spreading. Okay, those will be identified so we can develop a next generation of the vaccine and the drug. What about the new vaccine, new drug? We would like to target the gene under extremely negative selection, very conserved gene. So when we target it, it's not able to make a mutation escape the vaccine or drug. So that's the strategy we can develop to apply our theory to develop a new vaccine, new drug. So let's look at it some the potential application. So this is a gene from the SARS-CoV-2. You kind of can see at the gene level, it also follows the nature selection. You have some part, this is coding the 16 non-structural protein, and you see some protein are under the native selection. Typically, they are enzymes, they are critical for the viral function. And then you have some genes are under party selection, so they can mutate to improve the fitness of this virus. And we'll not go to biology. We don't have time to go to biology. Okay, so now let's look at the application. So everyone understanding the spike protein are the critical protein which on the surface virus for infection, which bind to the host cell receptor. And then we can look at its mutation rate change. Before we apply the vaccine, its rate is relatively low, but when we apply the vaccine, you see from here, this is 11 months of the pandemic start, you see it's under positive selection. Its substitution rates increased by 3-fold. So that basically means it's response to the vaccine. Okay, we can calculate the C/U relative substitution rate to find the mutation which give it increased fitness. Okay, where's that mutation? This top 10 mutation we narrow down. Okay, here you see that C/U is a very big number, bigger than 1. Okay, and then my student go to literature to check the mutation effect. And very interestingly, those mutation either increase its bonding with this called the human ACE2 receptor. This is responsible for the infection, or they reduce the bonding with this antibody, so that basically can help this virus spreading more. So either way, increase survival and reproduction spreading of this virus. So that explains the fitness increase. Okay, so those mutations should be included in the next generation of the vaccine. Okay, so now in US, the booster shot already start to roll out. Okay, and you also heard the drug remdesivir paloxivate, which targeting the SARS-CoV-2. So for remdesivir, it's targeting a RNA-dependent RNA polymerase. So here's the top mutation we found. Okay, so which basically you show is increased the fitness. So very interestingly, 2 of the mutation now already report drug resistance, and the other has not report. So we potential to evaluate the binding affinity to see if those mutations are reduce the drug efficacy. Okay, so the another paloxivate, which targeting a protease, and this is the top mutation we found, and so far no report drug resistance. Okay, you kind of can see from our method, we can ahead of time to predict some mutation may cause drug resistance, may cause the vaccine escape. Okay, so does this new theory can apply to the other variant? So this is the initial set of variants we examine: dengue, Ebola, measles, influenza, West Nile, Zika, Mumble, this set of variant. And then we collecting the genomic data, and then for this genomic substitution rate, we calculate its correlation coefficient because it's linear. And you can see coefficient is very high. This supporting is follow molecular clock. And we also examine the L distribution. Yes, all of these variables follow the L distribution. So we're confident all those variables follow our new theory. Okay, so let me go further. Because of this new evolution theory, is this new theory general for all the organisms, including the human? Well, this is my guess. I don't have data to back up my speculation. I would suggesting maybe balanced will be crossed out. So near neutral selection theory would be applied to all the organisms, including the human. And then what's the message got from here? And this is just like we survive in the society. You want work harder to improve your competitive advantage. And at the same time, you also want to have good luck. Okay, so both are important. Both are important. Okay, so now let's look at the dengue, because I know dengue is also a problem in Indonesia. And it's because the mosquito bite, and then people can have the fever and the other rash. And for the elderly, it's maybe can have the leading to the death. Okay, so this virus basically is coding by a genome which has some the structure, coding some structure protein, some the non-structure protein, and also has some the uncoding region, which UTR translate region. And So here we examine the L distribution. So remember this C over U, and you see its genome from the L distribution, and its translated region also follow the L distribution. And then for the untranslated region, used to be considered as the under neutral selection. Actually, it's not. It's also under natural selection. It's L distribution. And because we know now it has a critical regulation function. And remember, for the human genome, 98.5% of genome are not coding the protein, but they are very important for regulate the gene expression. Okay, and then you look at the number of mutation, and for the whole genome, it's look more like clock. But when you decompose into the genome, into the translated region and untranslated region, and you see it's under selection, particularly for the uncoding region. And if you decompose the polyprotein gene into the individual protein, and you see some gene under positive selection, some gene under the negative selection. Okay, so here's we summarize the relative substitution rate. Remember, 1 is boundary. So you have the gene under the negative selection, you have gene under positive selection. And then for the important gene, later we'll talk, this is basically the vaccine target. And you see already has some mutation, basically increased fitness of this variant. And then we also said for the current vaccine, it has a limitation, has so-called antibody-dependent enhancement effect. I will explain more in couple slides. And then because this is the dengue one, we know it also has the other dengue variant 1 to 4. And this is basically due to that the slightly change the variant and then original vaccine, the antibody-induced original vaccine will not efficiently bond to new variant. Okay, so now let's roll back a little bit about the vaccine development. So far there are 3 generations. Okay, so what's the first generation of vaccine? This is the first generation vaccine. It's called the live attenuated vaccine. So it's the virus. We want to remove some part of the gene, or we use a chemical to make it inactive. Okay, and the limitation for the first generation of vaccine is expensive due to the biological process. We basically have to grow this virus. Okay, and then this virus can sometimes have a safety issue and revert back to the virulent form. So that's the first generation. And the second generation especially use the peptide recombinate the protein. Okay, so they're much safe and much easier to produce. Okay, and so that's kind of the advance. Let's look at the third generation. Third generation is mRNA vaccine. Okay, it's literally just deliver the mRNA which coding the important viral protein, just like the SARS-CoV-2 vaccine from Moderna, from Pfizer. Okay, they're more efficient, they're rapid development, they're highly specific, they are very easy scale up the production, and they are very safe. Okay, so that's the third generation vaccine. Let's look at the dengue. So dengue, people typically use these 2 proteins as the antigen to develop the vaccine, E protein and then the membrane protein. So current vaccine is basically use that one. Current vaccine is the first generation vaccine. It's live attenuate vaccine. It's use yellow fever virus as backbone, and then use the membrane protein and the E protein from the 4 different strands and make this vaccine. However, this vaccine actually cannot apply to the people has not infected by this dengue virus, and because the antibody dependent effect. Okay, and what's the antibody effect? It's because the antibody developed through the vaccine are not strong enough to neutralize all the viral. And instead, they bring those into the cell, and then they make it more easy to replicate it, the virus, so makes the symptom much severe. Okay, so those are due to the mutation. So this is the issue with the viral mutation. Okay, so now let's think what's the key property if we want to define a perfect kind of vaccine. Okay, so we want high specificity, we want high selectivity, we want high sensitivity at molecular level. So specificity is that we have the epitope which are targeted by the antibody and only very specific, okay, and of the viral peptide. And selectivity basically means we only want targeting the viral protein. We don't want to have a human protein or other protein has the same epitope. So then it's not selective, we're targeting self. What means high sensitivity? We want the interaction between the antibody with the antigen as strong as possible, so that we bonding constant will be very, very small. Okay, so that's the idea vaccine we want develop. So now, can we use our new theory to help to find those kind of the antigen? Answer is possible. So here I calculate out the relative substitution rate for 4 dengue viral. And then we want conserved region. We want smallest value because those conserved region basically will be less vulnerable due to the mutation, cause the vaccine escape, cause the antibody-dependent effect. So here, the 4 black box is a good region for that. And then we can translate those regions into the protein structure, and then we can examine to find the region has a potential maximized interaction with the antibody. And then we also can search the protein sequence, make sure those regions will not be in the human genome. So in this way, we're able to find some the optimized antigen for the vaccine development. So this is we start to doing this kind of work. So hopefully we can show you some the result, experiment result very soon. Okay, so this is the part we optimize interaction. So E protein, which are the good conserved protein for vaccine development. And this is the antibody. So we want optimize potential interaction. Okay, so then what experimental validation for the new vaccine? Okay, so first we had validated, we have the messenger RNA, and then we infected the cell we want, we're able to see expression of the viral protein. Okay, so that's the first step. And the second step, we'll inject that mRNA into the rat so that we can check the immune response. Okay, so and if you are interested in the vaccine development, interested in this new area to apply the new evolution theory, you're welcome to join my group, or you contact with my collaborator, Dr. Alba, and we have the many years collaboration. Okay, so that's all my talk. Hopefully I within the time scale.
Okay, Professor Wu, thanks so much for very informative talk. Now come to the Q&A session. I invite participant both offliner or onliner to ask a question, maybe one or two. Oh, okay, please mention your name and your university when asking question. Yeah, please, committee, give microphone.
Yeah. Thank you so much for the opportunity. Dr. Wu, my name is Rudy. I'm from Indonesia, and I have a few questions here. I actually have three. We'll see if we have, if the time permits. The first one, as you say that there's always ideal condition that we want in us developing a vaccine. The conditions that I meant was the high specificity, and then high selectivity, and then high sensitivity. So my question would be, what percentage ideally should we use as the cutoff? Because I know many times when we're trying to develop a vaccine, then we are faced with the trade-off. When we want to increase the sensitivity, specificity goes down. So what would be the ideal cutoff for three of them? Should we do like 70, 70, 70, or should we do 80, 80, 80 if possible? That's my first question. And second, I really hope that you can share your screen again. There's one part when you showed us the like genome data set, I believe. I wonder when we are analyzing it, yeah. I think it was about like the
This one?
last. Oh, we're not seeing your. Oh,
Is this one?
No, not displayed.
yeah, we're not seeing your screen yet.
Oh, okay. So I need to share.
Yeah, the other screen.
Okay, so I need to share. Okay, so can you see the screen?
Yeah, we're seeing your screen now.
So which slide?
The top is showing RD, RP, and NSPS, remdesivir.
RDRV.
And,
Okay.
yeah, that one.
So you mean
There are
this
two
one?
boxes.
This one?
Yeah, that one. Yes.
Okay, good.
Yeah. So I'm curious about the bottom box right there. You said that one of the field that we can explore is the genomic data set. So my question would be, when we are analyzing the genomic data set, are we accounting for like differences in characteristics, like differences in demographics, for example? Because I know like these genomes are collected from humans. And that being said, different human would potentially have different sequences of genome. That's my understanding, and you can correct me if I'm wrong. Does that make sense? So I'm wondering, because I came from pharmacoepidemiology background, and we do studies with humans. So I'm just wondering if we can incorporate the human analysis aspect when we're doing any study with the genomic data set. I really hope that makes sense. If not, we can go to the third one. Otherwise, I can stop.
Okay, thank you, Dr. Rudy from Universitas
Dari Tadulako.
Tadulako. Okay.
Okay.
Thanks, Professor. Can you
Yeah.
get the point?
Yeah, let me.
The first one is ideal cutoff for specificity and sensitivity.
Yeah, okay. So let
And
me
the
answer
second
one
one,
by one. Yeah.
how
Yeah,
different human can induce different sequence maybe? And
Oh, in the analysis.
yeah. Okay, Prof. Wu?
Okay, so first, okay, the reality we always have the trade-off. So I think there's no kind of the set of rules that what's the ideal trade-off, and we want to put in practical kind of aspect. As long as that vaccine basically can save people, that vaccine can reduce the risk of the infection, then should be good. But one thing, remember, has not explored is that we so far not fully used the available information for ideal design. Have we used all the genomic information from the virus? Have we used the genomic information from the human? Have we used the structure information from all the protein we know? Have we used the interaction data we can generate it through the theoretic computation? We're not yet to use all those available information to optimize the vaccine, the drug we want to design. So I must say there are still big room to improve. Okay, so your second question is genomic. Genomic, I forgot. What's the genomic? Oh, okay. So it's the okay. Let me first answer this one. So here, what I mean, we need more genomic data. So one of the so surprising kind of finding for the SARS-CoV-2 is that because modernization, globalization, then the communication, the transportation, spreading, really it's just one village. You can't, from SARS-CoV-2, you can't see that. It's just start one place and then the whole world suffer the same things. So now I don't concern about that. I will including all those data into the analysis because now spreading is so fast because the modernization, globalization. Okay, so and then in this case, we want more genomic data because this drug just rolled out very recently. Remember, just this year. So then, if we want to look into the drug resistance mutation, we have to waiting for the more data to came out so we can analyze. And what's your second question? I forgot.
Yeah, about the genomic data set.
Okay, yeah, that's the question. You should include it. And because analysis is very cheap, so you can include it in the analysis, remove it for another analysis, and then to look for the empirical data to see which one make more sense. So that's my suggestion.
Okay. Yeah, thank you so much.
Okay, thank you, Dr. Rudy. Give applause to Dr. Rudy and also to Dr. Professor Chunhu. So, if anyone can ask question, one question maybe, and please monitor the onliners. Is there? Okay, anyone? Okay, Dr. Adrian.
Hello.
Hi.
Hello, can you hear me?
Yeah, I can hear.
Okay. Hi, I'm Adrian from Oleo University. I will not ask about the content of your topic, but I do appreciate for your great presentation. I will ask about our further collaboration, the possibility of our further collaboration after you have like collaboration with Professor Arba. So do you have like special plan or another plan to have a collaboration with Professor Arba or maybe for another researcher from our university?
Well,
okay, yeah.
Thank you.
I think it's clear.
I'm open, so I'm open, so you can talk more with the Dr. Alba. Dr. Alba, we have collaborating how many years, Alba? Okay, it's very long. And so I tell you a story, and Alba sent me an email to say we have an interesting question, and do we want to collaborate to do something? I said yes, let's start it. So then it's end up probably now we have the 10 publication together. So science, no boundary.
Okay, thank you, Prof. So the point is the collaboration is open for us, particularly Universitas Holyoke, to collaborate with Professor Chunhu at Rowan University. Okay, I think we are in the end of session. Yeah, so ladies and gentlemen, I would like to conclude our session today that the new evolution theory is developed by Professor Wu and his team. It can be used to investigate the substitution rate of other RNA virus in human, such as influenza, HIV, or dengue in Indonesia. So I think because it is new, so the perspective of this theory to be used in our research is, I think, very open. So I would like to thank Professor Kunwu for his sharing today, and also for the participant for very active participation. Finally, we give a big applause for the speaker and for you all. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
We give our deep gratitude for Professor Shunwu, PhD, and Professor Muhammad Arba for the amazing session. But before that, please, Professor Shunwu remain with us because the organizing committee would like to present certificate of appreciations to our speakers. Please be invited, Professor Arba, to present certificate virtually. Currently, the certificate of appreciation is being conducted by the Professor Arba to our speakers, Professor Shunwu, PhD. And please still remain with us, Professor Shunwu, because we will take virtual photos with the participant that is online. And please activate the camera and give your best smile for the participant in via Zoom meetings. All right? On my command, on 3, 2, 1. Thank you, incredible session. Please give a round of applause for once more for Professor Shunwu, PhD, and Professor Muhammad Arba.
See you later, Professor Wu.
Yeah. Thank you.
Dear honorable distinguished speakers, just ladies and gentlemen, we will continue to the next agenda. In this agenda, we will talk about the drug availability and the factors that affecting Indonesia and how to continue and how to measure and improve the performance. Who will be presented by the Professor Dr. Apothecary Satibi, and afterward the session will include the question and answer session of the topic. So, ladies and gentlemen, please be invited to the stage our moderator, Associate Professor Suryani. Before I hand over the session to Associate Professor Suryani, I would like to introduce our moderator. He is one of the lecturer of the Faculty of Pharmacy, Universitas Halu Oleo. Her research focused in the field of drug delivery system. Her educational history includes the Bachelor of Pharmacy degree at Universitas Islam Indonesia and continue her pharmacy profession education at Islamic University of Indonesia. His master's degree in pharmacy was pursued in Universitas Gadjah Mada. He has conducted several research projects in collaboration with the Department of Industry and Trades of South Sulawesi and also the Director of Human Resources and the Director General of Higher Education of the Ministry of Education and Culture, as well as the Research and Technology. And her complete profile you can read on the big screen. So, ladies and gentlemen, and all the participants, it's my honor now to hand over this session to Associate Professor Suryani.
Okay, thank you very much for our beautiful MC with their traditional dress. Good morning, ladies and gentlemen. Welcome back to the second session of the first Halu Oleo International Conference on Pharmacy, Health and Applied Science. My name is Suryani and I'm going to be moderator for this session. As the information that our presentation till the next 45 minutes, and the speaker will talk around 30 minutes. And after our speaker have presented his topic, we will have a discussion for about 15 minutes. And all the audience can ask the question. If you have a question, you can ask by raising your hand. And for the online participants, you can type your question on the chat column, and I'm going to with them for you. Our speaker today is Professor Dr. Apothecary Satibi, MSc. And the topic will be delivered by our speaker today is about analysis of drug availability and factor that affecting in Indonesia. Okay. And so I'm going to invite our speaker to join with me. Please welcome our speaker, Professor Satibi. And